So my name is Adrian Munoz, and I'm from the University of Arkansas, Fayetteville. And my title is Discovery of Putative SEP-D Signaling Network in ZMAs through a transcriptional lens. So for the past 10 weeks, my project has been computational, and much of this research I was looking into is we're looking into a known long distance nitrogen signaling pathway. And what this means is when a plant, particularly Arabidopsis, which is our model plant, is under nitrogen stress, these SEP genes or C-terminally encoded peptides will go from the root to the shoot to SEP-R, and then SEP-R starts producing SEP-Ds, which is downstream, and from shoot back down to the root, and it gives you the clues like, hey, we need more nitrogen. Can you start taking up more nitrogen? And so the nitrogen-rich soil will start taking up more nitrogen from the root to the shoot, back down to the shoot to the root, to the nitrogen-deficient soil. So there's already been much literature explored into Arabidopsis and also tomato, but both of these are dicots. And we wanted to look into monocots themselves, in particular Zea maize or maize itself and corn. And we wanted to look into these genes orthologous to Arabidopsis, and one way was to look at the putative genes of SEP-Ds themselves. And so bringing us to our background, just to give you more clarity, SEP-Ds are also known as GRXs, glutarodoxins, and GRXs are also known as ROXEs. So for the duration of my speech, I'm talking about, when I say SEP-Ds, I mean GRXs and also ROXEs. So hope you can keep up with me. And so one literature review looked at the entire GRX gene family that can be split up in 5 notable clades. But for our research, we looked into the beta gamma clade, which has all our ROXEs right here in dark blue. And so we wanted to know the orthologous maize genes of ROXEs and GRXs, comparing it from Arabidopsis. And so how we did this is I was given RNA-seq data from field trial experiments. And we also looked into public studies of literature reviews utilizing 409 plant species and also the greater GRX gene family with a focus on maize. And so my experimental design was looking at the gene expression using DSeq2 for normalization z-scores to see the expression of SEP-Ds under nitrogen stress. And from there, we applied multiple sequence alignment and phylogenetic trees to see how these cluster together. And so that brings us to our results, where we see the ROXEs and GRXs of Arabidopsis and maize clustered together and highlighted in purple. We see our genes orthologous to Arabidopsis that play a role in nitrogen stress on this long distance signaling pathway. And then highlighted in yellow are our ROXEs 6 through 9, which do have the ALWL motif right here. And then highlighted in blue are our ROXEs 11 through 15, which, I'm sorry, highlighted it in yellow are ROXEs 6 through 9, which do not have the ALWL motif. Highlighted in blue are ROXEs 11 through 15, which do have the ALWL motif right here. And so now we're looking into the maize genes orthologous to Arabidopsis, which share the closest relationship to playing a role to this long distance signaling pathway. And we do see expression right here clustering together. These genes are playing a role when they're induced under nitrogen stress. And we can say that this is genotype dependent. So it opens opportunities to look into these genes, but also look into the maize genes outside of the ortho group and see if they also play a role in this nitrogen stress and signaling pathway. And so it just opens up more opportunities to look into the research of these genes outside of the ortho group, as well as looking into the SEP genes are SEP-R and SEP-D, because it's all under SEP-D, or ROXEs, GRXs, and it just opens up more opportunities to look into different time points of maize, as well as different tissue types, because this is all under the leaf tissue. And we also had data on roots. So it just opens up a whole new PhD project for root, shoot, and leaf as well.